this. Okay. So the PowerPoint guide is this one right here. And when you open it, the last slide was that, that one in the notebook. And then there's this link here to the website I'm taking you to. Any questions on any of that? Okay. So the last section is visualization of the data that you obtained last time, or at least that type of data. Here's what we created, the middle, and let me know if there's a problem with sharing the screen. Somebody can let me know. So you guys can see the website, right? Okay, thank you. So what you get out of all of this for Callisto is a small object. Callisto remembers the thing that does the pseudo alignments and then counts of the data. So we have target ID on the left, which is your gene, a couple estimates of length, and then the most important thing are the counts. These are estimated counts here in the fourth column, and then in the fifth column, what are called TPM, transcripts per million, which are counts normalized for the length of the transcript. So the idea being that if some transcripts are longer, they may get more counts simply because they were longer and were randomly sequenced more just because they were longer. And so usually I don't worry about that, because as long as all the samples have genes that are the same length, it doesn't really matter. But some programs do want you to length normalize, and I think the one we're going to use today does. Okay. So Callisto, let's see. All right, so I'm going to just scroll down to the IGV Integrated Genome Viewer. Oops, that is not what I wanted to do. I'm going to scroll down to this web page where it okay, it takes us to this notebook preview, and then I have another scroll bar where I can scroll down a little ways to visualizing in IGV. The Integrated Genome Viewer is what I use for kind of looking at raw data. And so there's just 3 steps to the analysis here. Okay, the first thing is, well, I asked you last time to download the Integrated Genome Viewer, so hopefully you have it downloaded. If you don't, you're going to want to do that right now. And then once, I'm assuming that most people have done that. Once you have it downloaded, I have it on my desktop, for example, so I can show you my desktop. Mine's just right here, an icon, and I've already double-clicked it. And so for me, it opens up to something that looks like this. Maybe in the let's see. I want to try something a little different as far as your all's responses. So if you were able to install and now have opened the IGV, can you give me a, let's see, a thumbs up, or let's see, what else can you do? Okay, yeah, just give me a thumbs up or some type of icon on your screen. So I've got, see, those thumbs up only last a few seconds, but okay. So about half of you are still trying to work on it. Well, there's 3 steps that are mentioned in the web page. The first thing is to pull up the right genome, and this one we want is mouse version 10. And so for me, it's already pulled up, MM10 on the left here. You can see that there's a few human genomes, mouse, and then a rockfish genome that I use. This is the Ebola genome I'm using for a different class. anyways, the MM10, if you pull that up, it'll download information off the web, and you can see the chromosomes across the top without any information aligned, except for at the bottom. These are a density of genes along those different chromosomes. That's all it can show at this scale. Okay, the next thing that's in the website is to search for the gene leptin by putting in LEP inside this search bar here, LEP. So when you type in those letters, it'll bring up something that you can click on. So when you click on that and then hit go, oh, it's already taken me to it. This is a 13 KB window, and you can see the structure of the gene is down at the bottom, where there's looks like maybe a tiny exon on the left, or maybe this is part of the promoter. But this is the introns are shown with a thin line and then the arrows, and then the exons are the thicker bars, and this is probably the coding sequence with the even thicker bar on either side, and then the untranslated regions are the slightly less thick bars. Okay, what's something that's annoying me, you can see how every time I try to point at something, it opens a window. I'm just going to take my mouse up to this modify pop-up and say show details on click, not on hover. So that way I can point to something and it won't be annoying to me. Okay, so we have the genome. We've moved to the leptin gene, and now we want to load information from URL. And so here, if you go to file, file, and then load information from URL, you can paste in a URL link here. And so we can get that link from, if we just scroll down to step 3 here, from this data here. These are links that have what are called BAM files, which are alignments. I'm going to go ahead and choose the third one of the high-fat. So these are mice that were on high-fat diets and either had either control tissues or tumor tissues analyzed. I'm just going to go ahead and pick this third one, copy, and then go back to IGV and paste in that link. And then you just have to wait a few seconds. And it will, it's pasting in alignments in a BAM format to the entire genome. So it's a big file. You could search for other genes. Okay, and so mine, is there anyone having trouble with any of this so far? Maybe you could mention in the chat if you're having trouble. But for me, you can see some of this stuff aligned. I'm going to go ahead and change the name of this thing that just says pseudo alignments.bam. I'm going to change the name to the thing that I just copy and pasted from. So this was high fat diet control 3. So I'm just going to select that, copy it, and then go back to IGV and right click on the name of this alignments BAM file. And then when you right click, the very first option is to rename the track. So before I upload a second track, it's helpful to have it named, appropriately so you don't get confused later. And against that, I'm going to compare the high-fat diet tumor 3. So I'm just going to go down here and copy paste. Oops. While it's loading, I'm going to grab the name of that second file off the web. High fat diet tumor 3. And I tried to pick ones actually by my recollection that might show the effect a little bit stronger. So I'm cheating a little bit, but okay. So I've just downloaded or uploaded, well, who knows whether it's up or down. I guess download because it was on a server and now it's on my computer. 2 samples, the high fat diet control versus tumor. And at this point, well, why don't you guys tell me in the chat if you've been able to get this far, or how far you are if you have not been able to. Where are you? And I guess we're still missing Naomi and Eddie. Have you been able? Can you hear me now? Do you have audio, Eddie? I think you're muted. I could not hear you. Okay, you can hear us. So you can communicate, I guess, in the chat. So why don't you guys just let me know when you are able to upload those 2 samples and rename them, okay? In the chat, just let me know once you've been able to do that, and I'll just wait. If you need me to repeat any of the steps, you can just ask in the chat. Meanwhile, I'm just going to zoom in by dragging my mouse here and there. It's good to practice this so you'll have a better chance of remembering how to do it if you need it later on. Like if we wanted to check our own, the reads against our own transcripts, we could do that with IGV.
Can you hear me now?
Yep.
Okay.
So far I have, okay. Where'd that chat come from all of a sudden? Okay. Okay, so people are with me, okay.
Sorry, did you hear that?
Yes.
Oh, okay, yeah, sorry. I could hear you for a second, but I couldn't hear your response. I mean, yeah,
Okay.
I think everything's good now.
All right, I need to find a way to keep this Zoom chat from hiding.
Sorry, could you repeat quickly how you renamed the sequence once you copied it from the BAM file URL?
Yeah, I went over to the left side of the screen and I right-clicked on the generic name and then hit rename track.
Okay.
And then it gives you that option.
And we're doing high fat control diet.
Whatever one you copied over. Okay. Let's see. So a few other things that are nice about this, the IGV here. One is that these little gray bars on top. So I zoomed. You can zoom in and out a couple different ways. This little bar up at the top right allows you to hit a plus or minus to zoom in or out. And if you put your mouse over the gray histograms of coverage, these are coverage histograms for the reads. It tells you the counts of reads. So this one says total count 4, which makes sense because there's 1, 2, 3, 4 reads right below that. And that's the control. And then the experimental tumor sample has more like 30 in that same location. And you can see all these reads underneath that intersect with that position, which is a black dotted line. Okay. And if you, I put my note, I put show detail on hover again so we could see a little bit of information with each read. So shown are the read names, the length of the reads, and then the mapping quality. It says in the middle there, mapping equals primary at MapQ 255. So remember that Callisto doesn't actually align, it just pseudo aligns. So it really just knows where the read begins or ends. And so it does not give a mapping quality. 255 means unknown. But normally you'd look for like a value of 60, which would mean a really high quality mapping. A mapping quality of 0 would be would mean that it's aligned in more than one location. And if that were the case, that instead of a blue bar, you'd have a white bar, basically just an outline or hollow read. So for other types of aligners, you'll get the shading is important, but these are all going to be just unknown mapping qualities because they're not alignments, but rather pseudo alignments. Okay, let's see. So there's some other information there that let's see. Something else I just noticed. It says cigar 101M. That means that there's 101 bases that are either matches or mismatches, but directly without insertions or deletions. Remember that Callisto only looks at perfect matches and throws out any mismatches. So you do lose some data that way. Okay. Let's see. I just wanted to see if there's anything else in the that I wrote in the notebook that would be worth looking at here. Okay, I think that was about it for being able to look at your data. What do you think, guys? Does this look like, if you had a bunch of samples that look like the top and a bunch of samples that look like the bottom in 2 different groups, do you think that would probably be enough to call it differentially expressed? Or does this look like it could just be noise, or is impossible to tell? What happened to my data? Okay, I zoomed out so much that it hid the data to save memory. So let's see. How about the, so one has a lot more than the other. So if it was consistent, it would definitely be a significant difference. But why do you suppose the reads are distributed the way they are across the gene? Meaning that you have a big clump over here, and then these ones here seem to have lines between them. Does that make sense? And how so? Someone can do a verbal explanation. Why don't we see reads all the way across the gene for the one that's higher in abundance? You're going to need to unmute yourself if you have an answer. So those little blue bars are the reads. Recall that the exons are down below at the bottom, shown, the exon structure is shown below. So why don't we have reads that cover both exons and introns, the whole gene, instead of just exons? Somebody?
So does it have to do with the introns being removed and we're not reading the entire gene?
Right. The in an mRNA experiment, well, in an RNA-seq experiment, you typically sequence cDNAs that have been reverse transcribed from mRNAs. The mRNAs don't have the introns, and so yeah, we're just sequencing what cDNA is, not actual genomic DNA. So yeah, they're not there. And ours will look a little different if we were to plot it, because we are going to map against a transcriptome. So ours will be solid. The reads would be more solid. We don't have a genome for the brook trout. So if we wanted to view our genes on the IGV, they would not be all gappy like that. Good. All right. That makes sense? Okay. I guess the one last point here is that looks like some of these reads have been spliced, which means here's a read that was part one exon, and then another exon, although there's something weird going on with this one. It's really pretty colorful. Anyways, when you see reads that are connected across exons with a line, that means that it was a single read that covered the splice junction and has been connected bioinformatically, just to indicate that this is the same read that is on either side of the intron. Okay. And also the directionality of the reads is, in this case, from right to left, it looks like, because the arrow is pointing from right to left. So the sequencing was directional. They're all the reads are from right to left, it looks like, in that orientation. There's no red reads, which would be pointing the other way. Oh, I guess there's one, but it looks kind of like a messy one that might not be real. But if it was bidirectional sequencing, you'd expect reads in both directions and equal frequencies. So that is not the case here. All right, any questions about IGV? This is, what we did overall for GEA was get onto their website and run a quality trimmer called Fast QC, Trim-O-Matic. We looked at qualities using a program called FastQC. Then we did alignment of reads against the genome, and these are just sample alignments that have been pre-computed. But one of these is the one you guys did. And then visualize what that looks like, what differential expression essentially looks like in the IGV. And of course, in order to make this something much more useful, we'll run a program to determine the significance of differential expression at all the genes or across the genome. And that's what we're going to do next. Any questions on this, the IGV so far? We can use that later. All right, so I'm going to stop that recording.
Sorry, I do have a question though. I just couldn't get the audio to work. So my screen is like empty of all details where you have like, yeah, but where your mouse is right now, like that section is just completely blank. I'm not sure if I'm not zoomed in close enough or what the issue is.
Well, there's only 1, 2, 3, 4, 5, 6, 7, 8, 9. There's only 9 reads showing in the top sample. These were mice that were fed high-fat diets but were not tumor. And these ones on the bottom, where there's a ton more reads showing, all those blue bars, those were the high-fat diet tumor samples. And so those ones are expressing a lot more leptin. Each of the reads there is a random sequence of mRNA a pool in the samples. And so it is representative of there just being more mRNAs and more of that transcript type expressed in the cell. So up in the top there, this is essentially like our controls, where you expect it to be more empty. And the bottom one is a lot more full of reads.
Yeah, but what I mean is like both those panels are just empty. There is no reads, even though I loaded it.
Okay, so zoom in more.
Yeah.
and it might show, because
Yeah.
it
When I
doesn't,
try,
go ahead.
I was gonna say when I try to zoom in, it just gives me like the loading circle. Your mouse turns into a loading
Okay.
uh, circle, and it just doesn't do anything.
Well, I guess depending on your computer, you might have issues with speed or memory.
Gotcha.
Um, so that, I guess that's possible. There is a way to edit the preferences so that it'll only show you the reads if you have a very small amount of data showing, like zoomed into 1,000 base pairs or something, instead of, mine's at 10,000 right now. And I think the default is 30,000. So that's probably the problem, is that
Gotcha.
yours is a much larger default than mine. So
How
yeah,
do you? Sorry. Go
yeah,
ahead.
I can try to show you that. So let's see, view preferences, and then alignments. And then right here under view preferences alignments, it says visibility range threshold KB. If you change that to like 10, or even, let's just make it 5, and then hit save. So now it will just be blank unless you're zoomed into 5,000, a window of 5,000 nucleotides. So my window is 3,500, so I can see what all the reads. But if I were to zoom out, it would disappear.
Gotcha. Yeah, my looks like my screen is just dark now. So I'll try to change that for when I can reopen it, or at a different time.
Okay. Well, you can try zooming in like a really small area, like I just did there. This is just
Yeah,
40 base pairs.
what I mean is my IGV, like the application window is just like black now.
Okay. There is an online version as well. So maybe for next time, I'll give you the information about that version too, using a server instead of your own computer.
Gotcha.
Okay. Great. So any other questions on IGV right now?